So myself, Dr. Susmita Shukla, working as an associate professor at Amity Institute of Biotechnology in Amity University, India. So as I've already said, that I will be giving you certain glimpse of how the farming has been changed and how we have gone to the advanced farming. So here some of the techniques are there, which is related to the vertical farming, then CRISPR gene editing, which is already there, which I've already seen, and the precision agriculture and hydroponics. So people are now going for different vertical farming as well, the CRISPR gene editing, which I've said, hydroponics, not only in the bigger farmland, but they have also started in a very smaller phase. Even we can see that people are moving towards the kitchen garden also. Then kitchen vertical farming, even I have seen that our students have developed a very small hydroponic system, which we can keep it in our kitchen, and we can have the green veggies in the kitchen itself. So that is how people are going towards innovation, and different techniques are coming through it. Now, as I've said that we are promoting sustainable plant growth. So here you can see that I have also seen yesterday's speech of some of the speakers who were well defined how they have reduced the pesticide use by their different biotechnological tools and different science modification which we have done. So reducing pesticide use has been, again, it is for the sustainable plant growth, which we are targeting to reduce the use of all the different chemicals. Then moving to the support local food system, you can see that even we have started working, many are working. Their local food system are so rich that people must know about those food system and should use it for the different permacultures. So, and then the different non-toxic farming practices are there where we are totally going towards the organic. So this is how we are going, and we are dreaming about the sustainability. So when we talk about sustainability, we say that it efforts to reduce the use of pesticides, fertilizer, conserve water, promote biodiversity, and different innovative approaches which have been utilized. So regenerative agriculture, which emphasizes soil health and biodiversity to improve the long-term productivity. So these are the different terminology which has been used to get the sustainability. And even for the agro biodiversity, we have worked on different trees and crops, and even the livestock system to enhance the biodiversity which has been seen. Here we go. When we talk about the role of plant science, it's a very crucial role which is achieving. As I've just heard the before speaker who was saying that without plants, we are nothing, right? Plants are there, so we are there. If no plants, then no population in the world. So it is having a very crucial role there. About the sustainability goal, if we say that we should be knowing, the plant scientists are knowing the different physiological and genetic, they are going through different studies. Even recently, they have started working on the different plant species which they are targeting to reach towards different planets as well, not only Earth. So they are trying to go for that. And even for the genetic engineering also is having its own importance, where people are trying to raise the biotic and abiotic stress resistant plant system, the different varieties they want to raise. And we are a small part which we are also involved in that. Secondly, the bioremediation use plants to clean up, which I have heard yesterday where Dr. Priyanka was talking about this bioremediation as well. And she spoke very well how the heavy toxic metals can be utilized by the different microbiome analysis, and it can be done, and to understand how it can be improved and can be restored in the ecosystem. Now, this was just the glimpse which I have given why and what way we are going towards a greener future. As we know that greenery, we are targeting for everything. Even now the meat which we are targeting different plant proteins to make that type of things. So what our work, our lab is involved in, basically, we are into the plant tissue culture. We have developed different protocols. Now we are already having 10 protocols in our hand. Whenever it is being asked, we are ready to deliver that. So here you can see that how and what way we have been utilizing it. So plant tissue culture is a very important technique which has been used for producing the disease-free plants for not only the different crops, but basically it has been given emphasis on the horticulture. So propagation of rare and endangered plant species is also has been a very important part, because what we have seen that whenever uh, as of India, we say we are very rich in these kind of germplasm which we are having, but still there are, due to some of the climatic condition or different parameters which affected erosion and all which I've talked about, many of the plants are phased or fall into the category of extinction, and they have been listed in the red listed. They are being red listed, and many have been put in the endangered categories. So being a tissue culturist, it is our aim that we should be taking care of this and increase the gene pool, not only increase the gene pool, but to protect the biodiversity as well, to protect the germplasm as well, to conserve the germplasm. So with this respect, I will be showcasing some of the work which we have done in our lab, along with my collaborators and my research scholars. So I will be showing you how and what way we have worked on that. Here is the example which has been given where the clonal propagation of the banana has been given. And if we talk about banana, we are the highest, we are the one who are producing maximum banana, and even we are transporting and exporting to the other countries as well. The conventional cloning methods, and the important thing what has happened, that when we were producing these kind of bananas in the conventional means, it was giving the food and the food products which we were getting about more than 12 months. But later on, when we were successfully got the protocol related to the clonal propagation of banana, it has been seen that the time has been reduced from 12 months to 9 months. So that is the best important part. One is disease free. One is we can multiply the plant as many times as we want. We can get the clonal progenies. And at the same time, we have reduced the shelf life or the fruiting life of the plant. So this was about the banana, and we have started working on the species, as I've already said in my previous slides, that we should know and realize the potential of the food crops or the different plant species which is there, which are having their own importance. We should know them, and we should try to increase those production for the different importance of it. So we have started working on the indigenous banana, where we have selected 4-5 indigenous banana species in our country, and we have successfully propagated that. So the whole terminology I'm not saying, the simple methodology for the propagation is being given through this schematic representation. So here you can see that the mother plant selection is very, very important. If the mother plant is stronger and healthier, the progenies will be the same. So similarly, from the source plant, the suckers has been taken, and then after sterilization, the primary treatment, it is being transferred to the different media where the multiplication, shoot proliferation, root proliferation, and the hardening take place. And finally, we are able to get the bananas. So most of the thing you can see that how it has been spreaded so far, and we have become the most popular. What you can see that we are having. Just a second. Sorry. So we are having the most popular species in our hand. So here you can see that how we have worked on. This is another horticulture crop which people has worked on, and even we have worked on a very important species of pomegranate, that is Fullea abrupta. That is one of the important species in India which we are having, and we are the one who have successfully propagated it. Even we have patented the different things which are there, and it is readily available with us. So why pomegranate? You know that it is a full Punica granatum which is there, and this is the first 5 cultivated food in worldwide. So there are different varieties, there are different things which are there well suited for that. So here some of the glimpse has been given, which is talking about the agrobacterium rhizogenes and transformation and the extraction of phytochemicals and all, and how it has been used for the therapeutic value. So these are the different technologies which are being applied and which we have successfully done in this particular lab of ours. Now, another important part is the one which is the olive. And we can see that olive are growing throughout in Italy, Spain, Israel. And now we are also one of them where we are producing the genetically uniform and free of the major disease plant. So this we have successfully got it. And why this was done? Because it is a high economical value, and if we go and calculate the different amount and different things, we will be able to understand that what are the use of olives. Though people of here are not much aware of it, but yes, it has been started creating its own interest among the youngsters who are every time looking forward for these kind of foods. So this is just example has been given where in Italy, major olive growing countries has been counted and more than 24 commercial micropropagation laboratories has been set up for this. So the similarly, it is having its own importance and throughout the worldwide, it has its own way. So moving to the another variety which we have worked on, that is the citrus, and not only the normal citrus which we have worked on, we have worked on the endangered citrus of the region of India. That is very, very important, which is there. So why citrus? Again, it is an horticulture crop. It is having its own importance. The species which we have selected for the propagation part was not only for the edible purpose. Right? So its green is actually, it is having its own medicinal values as well. It is having a high therapeutic value, and basically it is being used for the neurological disorders, which we are targeting to find out the different phytochemicals which are present in that particular citrus and how that can be implemented for curing and for protecting the different neurological disorders, specifically the Alzheimer's and all, which we are checking on the different models. So that is a different part through the translational research we are working on. So here you can see how it has been already worked on and how it has been gone. If we go for this kind of propagation, we need to know much more about it, right? So we have to go for biodiversity conservation, which I've already said. We need to collect it. Even if we are propagated, we should submit to the seed banks, in vitro conservation and field collection. These are already there. We ourselves has implemented and we have planted these endangered citrus plants in the campus of Amity University itself. So here, as I've already said, that we are also working on the role of plant tissue culture in transgenics. So this is what we have selected. We have started working on the Vigna species. So these are the different species which are there. These are our own work and we have given for publications also. We have communicated it. So here, what is the thing which has been seen? This is a glimpse of the work where the plant transformation has been directed, shown, and by the direct and indirect method, we got successful regeneration and transformation. So we got the success in transforming the drought resistant gene, that is the DRED. So DRED gene has been successfully transferred there, and we are in the phase of, I want to say, the different analysis of the trait analysis we are going with. So these are the different thing which is there. And tissue culture is the pathway for raising a transgenic plantlets. Without tissue culture, we can't think of transgenic. So if the tissue culture protocol is already ready, then it is again a plus point for the transgenic crop also, because what has been seen that after transformation, the propagation is difficult. So that is why it is very, very important that we should be going with this plant tissue culture first, technology development, and then we should be going for the status. Here, I'm showcasing the work which we have done. So this is a robust micropropagation protocol, which I've already talked about the Punica granatum, which we have worked on, and this is a variety known as Pulayarantha. You can see our team members are there. Dr. Vidya is there, and Ms. Dharmala Raman is the my PhD scholar who received her award in 2020. And during that particular lockdown period, we have worked on and we got success in these kind of propagation. So what has been done? Those mother plants were selected, the healthier one. And from that, we have tried for many different explants, not only the nodes, the leaves, the roots, everything we have tried because we were finding difficulty in propagating or getting to get relief from the contamination. So again, we have modified our technology, the protocol which we were working on, and then we have started with the different sterilization of the explant preparation. Now, finally, after that, we got the nodal segment to which we have worked on. So here the sterilization and explant preparation is there where you can see the we got after the formulation and the standardization of surface sterilization, we selected the nodal segment, right? And after the nodal segment has been given, you can see how the bud break took place and multiple shoot proliferation affected. So there we tried different medias, not only the MS media, but we have tried for WPM media as well. So you can see how WPM plus BAP plus PVP has been given the maximum amount of shoot proliferation. So you can see that more than 5-6 shoots, and even we got the maximum 8 shoots at a time, multiple shoots which we were getting, and this was successfully standardized in this particular media. But again, I would say that it may differ from species to species. Then finally, we went with the routing and the handling. But during this particular step, again, we faced lot many obstacle in the hardening system. So that we have done by the shock treatment which we have given to the during the routing system, where we have put in the concentrated NAA, and then we have transferred into the different substrate. And not only this, we have also done the different genetic fidelity tests to understand that all the progenies, all the in vitro plants which we got from the mother plant are of similar type, are of uniform type. So we went with the RAPD and ISSR marker, and we got the very result, yes, they were true type. And finally, it was planted into the field, and still we are having it has been grown up, and now it has started fruiting as well. The next part, which I have said about the olive, this is the work which we have done. So olive, again, it was a very difficult plant, and you can see that trees are very difficult to handle with the tissue culture techniques. So here again, we went with different kind of techniques which we applied for. And here also we went for the different explant selection, different sterilization methods, which you will be able to see in my paper where we have published about the micropropagation of Polio europaea barnea, where we have worked on. And certain thing was there which we have added, supplemented, and finally we got the result where we successfully got the whole protocol. To get the whole protocol, we took about 3 to 4 years. It was not easier for us to get it because every time we used to find some of the other difficulties, either in the during the subculturing or during the hardening time. We were facing a lot of difficulty there. So here what we have selected, again, notes from the adventitious shoots were selected. That is the major part which we have seen, which we have disclosed, that not the nodes from the general or the other one worked. The nodes which we took from the adventitious shoot, that worked, and that gave the the shoot induction took place and the bud break took place. And later on, we transferred into the multiple shoot proliferation media, which was comprising of half MS, half Rubini olive media, plus Z8 1 mg per liter. And along with that, we have also added silver nitrate per liter. And this was successfully views, and finally we have transferred to the rooting, and finally it went for the learning. And simple genetics fidelity test we have carried forward, which we have said. Now moving towards the other one, which I've also talked about, that we have worked on the endangered citrus species as well. This is the glimpse of the indirect organogenesis, which we got from the thallus, and for there, because we were not getting here, many citrus you can see, they are also prone to the different endophytic microbes, which is very difficult to handle it. And those microbes keeps on coming. So we need to know and we need to stop those entries. So we did many experiments to understand the thing that we have done. And even from the callus, we were successfully got the regeneration part. You can see how the shoots are coming out from the callus itself. So this was a very good development which we got from the citrus plant, the indirect organogenesis. You can see for that we have selected the explant, which was seed and leaf. And after that we got the callus, and from callus different medias were tried, different adjuvants were tried, different phytohormones were tried, and ultimately we could standardize the effect of different media and the hormones, and we went for the shoot proliferation, and finally we reached to the rooting and hardening media. So this is again a different story which we have done. So I just want to communicate that working with a particular plant species is not an easy task. For working with a single species, it takes lots of time, as the result we will be able to know only after 4 weeks, right? So after a month, we can understand that yes, we are going to get some positive result or not. But if we are getting one time, it is very difficult. But later on, once you have standardized, it becomes easier. Next time you can propagate, you can grow as many as you can. That's what I was talking about, the sustainability, that how this tissue culture technique is applicable for the sustainability as well. Here again, you can see that we have standardized, as I was talking about, that we have worked on the indigenous banana also, and we have published this in the recent journal of Environment and Pollution Research, where you can see we have talked about the impact of sterilants on culture establishment. So that is very, very important step which we have to take care of. Without the sterilization, without the standardization of sterilant, which sterilant you are using, it is very difficult to go with the further stages of micropropagation. So that is why we have given the glimpse how and what we could do it, we could protect it. Because previously we were facing many problems, the contamination, then the leaching, browning, many things. So we have to go for combination and different combination and different trials of different experiments with different sterilants we worked, and finally we reached to a conclusion that yes, we finalize the protocol, which one is the best for the culture establishment. So this we have already published in a paper, which you can go for. Moving to the next, as I've said, that the work which we do is quite a typical one, means it requires time. So she is Dr. Taramala Raman. Now she has been awarded her doctorate degree, and we worked on the micropropagation of Punica granatum and Olea europea, and successfully now she has joined a particular university as a consultant, and she is doing a very good job over there. Here again, my different another PhD scholar is working on the different research which we are working on, establishment of in vitro regeneration and aggravated mediated genetic transformation system in Vigna species. So again, we are getting very good leads on that. Next, if we go with this is Mr. Tapiya Singh, who is now, I've sent her to work for future collaboration in Canada. And finally, when she will come back, she will be submitting her thesis as well. So we are working on the in vitro regeneration of citrus amount fruits and evaluation of its neuroprotective effect. Because I've said that, yes, neurological disorders can be cured, but certain chemicals have been found which was giving a positive impact on the different models which we have tried for. She is Miss Rutambra Bhutani, and again, she is working on the different indigenous Musa variety. And this is a very good glimpse of the work, part of work which we have done. We have got the different four varieties of India which we successfully got, not only from the northeast region, but from the different part of India. We collected those germplasm and we successfully propagated it. So these are some of the collaborators which we have seen and which we have worked on. We received funding from Department of Biotechnology, Government of India, where we were working on the embryo rescue technology and so forth. So now, if we go for the challenges and opportunity, as I've started my talk with that, challenges is there, climate change, environment degradation, food security is there. So that is why we can promise, we can say that yes, by doing these kind of work, we can improve, we can protect, we can be a part of overcoming these kind of challenges. Opportunities are there depending on the adoption of new technologies, because as you know that there are many different things which are happening in the field of plant tissue culture as well. So market demand is there, people are moving towards and they want these kind of things. And we are having many different organization in India. If you can see that we are having more than 100 companies who are doing the plant tissue culture techniques. So propagating the different plants through micropropagation technology. So these are the different thing which is there. And if we go for the future prospect, you know, those propagated plants, we need not to go and destroy the nature. We are having everything in our hand in the lab, and from that we can utilize those phytochemicals which are having different properties as say antimicrobial property, or which are having the properties of getting some kind of herbal drugs and medicines. We can go for the modern agriculture, the protected land farming and all. And finally, the commercial products like bioplastic, disposable cutlery, even the soap, shampoo, face wash, etc. we are getting from these kind of things. By having this, you know, that we are targeting the plants which are disease-free, and the amount of raw material which is required can be obtained anytime. So with this, I come to an end of my talk. So thank you so much. So if you have any question, please let me know. I will be happy to answer.